All right, let's get started. Good afternoon, everyone. I'm Weijia Hong, and on behalf of Larry Zapersky, who couldn't be here, I'm pleased to introduce the speaker for today's CTSI seminar in Churchland. Anne is currently the Arnold Chabelle Chair for Brain Research and Professor of Neurobiology here at UCLA. Over the past 25 years, and has made extraordinary discoveries and pushed the boundary of what we know about the brain. Anne obtained her PhD at UCSF with Steve Liesberger, and then pursued her postdoctoral work with Mike Shadlin at the University of Washington. During her postdoc, she made important discoveries about perceptual decision-making in primates, finding that the variance of neuronal firing rates serves as a crucial signature of neurocomputation during decision-making. She started her own lab at the Cold Spring Harbor Lab in 2010, where she rose from assistant to full professor and chaired the neuroscience program. In 2020, she moved her lab to UCLA, bringing her groundbreaking research program to our campus. And all of us couldn't be more excited to have her as part of our community. In her lab, Anne has made fundamental discoveries that has transformed our understanding of the neural mechanisms underlying decision-making. She discovered that the neural activities throughout cortex during decision-making is dominated by an individual's movements, especially any instructed movements that are not required for the instructed goal in a behavioral task. This finding has reshaped how we think about what the brain is doing during a goal-directed behavior. She also found that different cortical cell types drive functionally distinct activity patterns during decision-making and giving us new insights into how different types of neurons contribute to complex cognitive processes. Collectively, her work has provided profound insights into the neural basis of how we behave and make decisions in a complex world. Anne's scientific impact extends far beyond her own lab. She co-founded the International Brain Lab, an ambitious global collaborations that brings together neuroscientists from around the world to tackle some of the brain's most fundamental questions. This is not just about sharing data, it's about reimagining how we could do neuroscience on a global scale. Anne's work has earned her many awards and recognitions. She received the McKnight Scholar Award, the Pew Scholar Award, and just this year, the highly prestigious Praedel Research Award from the National Academy of Sciences, which is presented annually to recognize someone who has made extraordinary contributions to our understanding of the nervous system. What makes Anne special is not just her scientific contributions, but her commitment to advancing inclusion and supporting everyone in the neuroscience community. She creates Anne's List, a comprehensive database of female computation and system neuroscientists. It's now used by thousands of researchers worldwide, helping achieve gender balance in conferences and hiring around the world. Anne is currently funded by the NIH, NSF, and the Simons Foundation, and her lab at UCLA continues to uncover the important mechanisms that underlie decision-making and behavior. Today, she will share new insights from her remarkable work, and please join me in giving her a warm welcome to the stage. Thank you.
Wow, thank you so much for that lovely introduction, Weijia. I really appreciate it. Can everybody hear me okay? How's the sound in the back? Good, okay. All right, well, thanks very much for all of you for being here today. I'm gonna talk today about the intersection of vision and movements in the mammalian brain. So in my lab, we're really interested in decision-making, and we mostly study decisions in the context of movements. And we do that for a couple of reasons. The first one is that studying decisions in the context of movements is just kind of a natural thing to do. So suppose I made the decision that I wanted to walk into this building here at UCLA, that would naturally give rise to a series of movements, walking steps, opening the door, and so on, that would allow me to enact that decision and go into the building. And the other reason we study decisions in the context of movements is because it's a really convenient thing to do from the point of view of doing an experiment with a human or animal subject. So suppose you've trained, like we have here, a rat to report its decisions by making a movement to a left or right reward port. We can use this setup to track a series of left and right decisions over the course of an experimental session, keeping track of both the decisions that the animal has made and also interesting metrics associated with those decisions, like how long it took to make the choice and so on. So although we've thought about decisions in the context of movements for a long time, we decided to go a lot more deeply on this, to think both about the kinds of instructed movements that we train animals to make as part of a behavioral task, the uninstructed movements that they make spontaneously without any instruction from us at all. And we wondered about the inputs of both of these kinds of movements on neural activity and also on decision making. And thinking in this way kind of has taught, I think, taught us three things. And that's what I want to tell you about today. So the first thing is that neuroscientists have thought deeply about how instructed movements, those are the ones we teach the animal to do, impact neural activity, but uninstructed movements have received less attention. Secondly, that uninstructed movements activate diverse neural structures in mice. And finally, that I believe a new approach is needed to understand how movements can sculpt neural activity to benefit vision in diverse species. So I'm going to start here talking about the instructed and uninstructed movements. So people have thought about instructed movements that are trained to a human or animal subject as part of a task for quite a long time. And they've come up with a number of solutions that have hopefully served to ensure that movements can't account for neural activity differences. Okay, well, what does that mean? So for example, when I was a postdoctoral fellow at the University of Washington, I studied non-human primates making 2- and 4-choice decisions. And without going into too much detail, I discovered some intriguing firing rate differences in neural activity between these 2- and 4-choice decisions. So I worried that perhaps those neural activity differences were driven by movement differences between these two conditions. And specifically here, we instructed animals to make a saccade to report their choice about motion direction in the stimulus, and to ensure that differences in saccade metrics couldn't account for the neural activity, we simply looked at the saccades and we compared for 2- and 4-choice decisions, the saccade amplitude, velocity, and also its endpoint position. And by confirming that the movements, that these instructed movements were similar for 2- and 4-choice decisions, that gave us confidence to interpret the other kinds of neural activity differences that we observed. I should say that this is only one of a number of strategies that people have used to help them disentangle instructed movements from neural activity. I've mentioned a couple more at the bottom, including a particularly nice one that was led by James Bisley, who perhaps is not here, but came up with a really lovely way of disentangling those two signals in the data. Another way to think about this solution that's very much of the same vein of assuring that movements can't account for neural activity differences is one that I started to use when I started my independent lab at Cold Spring Harbor Laboratory. I had switched to a new model system studying decision-making in rodents, and I trained rodents, just like in the movie you saw a few slides ago, to either go to a left or right reward port to receive a liquid reward if that port corresponded to the correct decision. So when we tracked the animal's heading angle on left versus right decisions around the time of the movement that the animal was going to the port, they were of course very different. That's the whole idea. They were going to the left or the right. But when we looked earlier during decision formation, a time at which there were really interesting things happening in the neural activity, we found that left and right choices were not very different at all in the movements that they drove in heading angle. And this gave us some ease in interpreting neural activity changes between left and right choices during that decision formation period. And this is work that was published from my lab quite a long time ago. But the truth is that critters move a lot. And so even though as a field, we've come up with a lot of thoughtful ways to disentangle neural activity related to cognitive signals for neural activity related to movements, the truth is that animals are just moving all of the time. And they're not only making those instructed movements that we trained them to make as part of the task, they're also making a lot of spontaneous, uninstructed movements as well. This is certainly true of our rodent subjects, and you can see a lot of examples here. It's also true of our humans, any human really. It doesn't even need to be a subject in the experiment. And kids, for example, engage in a lot of chair rocking, despite the fact that this comes at a very high social cost, especially from the teacher, who usually gets very angry. So these movements are ubiquitous, and they occur even when there's a great cost to them occurring. So one thing you might be worrying about here, at least for the mice, is, well, maybe the reason these mice are fidgeting around a lot is just because mice are kind of ill-suited to do the kind of precise psychophysical experiments that have been the focus of the strategies for disentangling movements and cognition more generally. But the possibility that mice are just easily distracted, it really can't be the explanation. And we know that because mice make stable and reliable decisions despite frequent fidgeting. So for example, in a recent collaboration I was part of that Weijia mentioned, the International Brain Laboratory, we developed a mouse decision-making task where they use a little Lego wheel to report their choices. The wheel is coupled to the stimulus on the screen so that when the wheel moves, the stimulus moves too, and they use this apparatus to report decisions about the spatial location of a visual stimulus. And if they move the wheel the wrong way, then they don't get rewarded. We use standardized instrumentation to study behavior on this task. And I'll show you now some behavioral data that was led by 3 folks in my lab, Felicia Davatoha, Marsa Tahari, and Anup Kanal. And they, to visualize the data, plot the proportion of rightwards choices on the vertical axis as a function of the contrast of the stimulus. When the contrast is 0, that means that the stimulus is invisible, and contrasts close to 0 are very hard to see, and minus 100 and 100 are the very highest contrasts, since that's when the animal has perfect performance, either always choosing right or never choosing right, depending on what side the stimulus is on. We refer to this as a psychometric function, and this is a really good psychometric function in the sense that it has high sensitivity, it's very steep, and also a low lapse rate. At the very easiest stimulus intensities, the animal makes almost no mistakes. Here are 3 more examples from other mice that were trained by Felicia. And you might be a bit skeptical here, perhaps wondering whether these psychometric functions reflect not so much the ability of mice to do complicated psychophysical tasks, but instead the ability of experimenters in my lab to train mice effectively. And indeed, all these mice were trained at UCLA. But rest assured, here's a mouse that was trained at Berkeley. I would say it's not quite as good as UCLA, but it's pretty damn close. Here's one from NYU, from Cold Spring Harbor, and so on. And we actually had 8 different experimental sites and demonstrated that the behavior was reproducible across all of these different laboratories. So I hope this kind of data will put your mind at rest that distraction alone can't be the whole explanation, because our experimental subjects, the mice, make stable and reliable decisions despite frequent fidgeting. Okay. So I've told you so far, hopefully convinced you that neuroscientists have thought deeply about how instructed movements impact neural activity, but uninstructed movements have received much less attention, even though these movements in mice and definitely in children are frequent. Okay, so having made this observation, a couple of scientists in my lab wanted to tangle the second issue. What is the impact of these uninstructed movements on neural activity? And this work was led by Simon Musil and Matt Kaufman when they were postdocs in my lab. Simon is now at the Biozentrum Jülich in Germany, and Matt is at the University of Chicago. They're both assistant professors. And they trained up head-fixed mice to do a relatively simple task where the mice simply have to make a decision about whether the left or right side has more sensory events. So in this trial, there are more events on the left than the right. These can be auditory clicks, visual flashes, or both together. So the animal would be rewarded for licking the left spout. This is a more difficult trial because there are some distractor events on the other side, but the correct answer is still the left because there are more events on the left. This would be an impossible trial because there are equal numbers of events on both sides. And then just to round things out, this would be a trial where the correct choice was to go to the right. So this is a video of what the task looks like. You're seeing the mouse from the bottom up. That's its little nose and whiskers at the top of the screen. And at the bottom of the screen are 2 handles that the animal has to make contact with to initiate a trial. There's then a stimulus period, and you're seeing the animal now from the back. That's its tail hanging down at the bottom of the image. And in this visual trial, there's a visual stimulus that is presented. Here's the original view where the animal will make its choice. I'll show you this one more time. You can see that both of the spouts move in and the animal makes a choice to lick one of them, and then the other spout moves out of the way. That's just to prevent the animal from changing its mind. So Simon and Matt were curious about this question about the impact of uninstructed movements on neural activity. And to get to the bottom of it, they wanted to look at neural activity really quite broadly. So they brought in a technique that was at the time a new one for my lab called wide field imaging. And this allows us to look at the entire dorsal cortex of the mouse simultaneously through an intact skull. And to do this, we use mice that express genetically encoded calcium indicators in all of their excitatory neurons. We do that via the EMX-Cre line. And for anyone who's new to this sort of approach, the gist of it is that when a neuron fires, it will turn green and emit photons, and then we can catch those photons with the camera that's at the top of our setup. This technique turns out to have a lot of advantages. It's not a method for imaging single neuron activity, and that is a problem that we're going to return to later, but it has many advantages. Being able to image through an intact skull means that we can image lots and lots of animals, many more so than if we had to put in cranial windows. It also means we can image longitudinally, gain some insights into learning, and so on. So this turned out to be a really useful technique. A project scientist in my lab, Joao Couto, was the first author on a protocol paper we wrote about this approach, if you're interested in giving it a shot. We actually all worked together to build these setups. This is a sped up movie of a setup that we still use in the lab, although we had to disassemble it and reassemble it when we moved from Cold Spring Harbor. And a crew of 4 or 5 people working together can assemble the microscope in about a day and a half. And this is what the data looks like. So I'm going to show you now 2 videos of the same mouse. They're just from different camera angles alongside the neural activity from the wide field imaging scope. And they're slowed down by about a factor of 5. So you can see the movements and neural activity slowly emerging together. When we first started to look at this data as it came in, there were 2 things that really jumped out at us. The first thing is that the neural activity, which is indicated by that scale bar on the far right, yellow means more neural activity, that it was fairly widespread. This was surprising to us. We didn't initially think so many neural structures would be recruited for such a simple audiovisual task. And the other thing that jumped out at us was that the activity was highly variable trial to trial. And that surprised us because these decisions aren't that different from each other. Sometimes the mouse goes right, sometimes left, sometimes auditory, sometimes visual. But those small differences didn't necessarily lead us to believe that the single trial activity would be so incredibly variable. We wanted to get to the bottom of this, but since this was a new measurement approach for the lab, we decided that the first thing we would do would be to validate that these measurements made sense. So we first looked at the trial averaged activity, so the delta F over F, that's the fluorescence signal, it's a proxy for firing rate, as a function of time on the horizontal axis. And we compared groups of trials where the stimulus was on the right side, that's the contralateral side to the plot that I've shown you here, or the left, which is the ipsilateral side. And of course, as you know, in primary visual cortex, it's mostly driven by inputs from the contralateral hemifield, and that's exactly what we saw. The gray line is above the red line, exactly as it should be, and it had the known temporal dynamics that have been reported from primary visual cortex again and again. So this was great, and it validated that our measurement was doing what it was supposed to do. We measure the right signals in the right place at the right time. But this trial averaged activity, it won't answer the question about which signals, instructed or uninstructed movements or task variables, are dominating neural activity. And to get to that, we really needed to go to single trials. So the goal of Matt and Simon here was to build a model that would allow us to account for single trial activity in every pixel of the dorsal cortex that we measured. So for example, if this is trial 1, trial 2, and so on, we wanted to build a model that we could take held out trials and predict what they're going to do on every trial. That is, predict all those little wiggles that you see there. And when Matt and Simon first mentioned this, I felt it was a little overly ambitious in terms of a goal, because cortical activity is famously variable trial to trial. So I wasn't sure this was necessarily a really good goal. But they pointed out, they said, well, look, there's a lot of things that change trial to trial that could account for these fluctuations. There's the licks. Those are under the animal's control. They're not the same every trial. The stimulus comes on at different times because that's part of the experimental design. Handle grabs are different. And we also had collected video, this turned out to be super important, that might account for some of these fluctuations as well. So just to return to the nomenclature before, the handle, grab, and licks are instructed movements. The onset of the stimulus is a task variable, something that we built into the design, and much of the information in the video are uninstructed movements. This is slightly imprecise. We actually took a lot of steps to separate all of the video information from the other variables. And if you're interested in that, I'd be more than happy to talk about it in the discussion. For now, we put all of these variables together in a big design matrix and used L2 regression to pick the weights that best related these regressors to the neural activity. You might perhaps be troubled by the fact that these regressors are really of two different types. These are event-based regressors. These are what we call filterless analog predictors because they're continuous. And that's another topic that would be great to cover in the discussion if you're concerned. We thought about it a lot. For now, direct your attention to the right side of the screen, and I'm going to overlay the predicted activity for these 3 trials, which were held out from training. And you can see those now in red. So we were able to really capture quite a lot of the wiggles in these single trials with our model. I know this is a skeptical audience that would not be convinced by only 3 trials. So for the sake of completeness, I've shown you for averaged across all animals and all trials and all pixels in the brain. This shows you how much single trial variance we were able to account for. It's about between 35 and 40%. So we were pleased with that. And moreover, having a model that was working pretty well gave us a really important opportunity, which was to look under the hood of the model and see which variables are driving this fit and gain some insight into this question about instructed and uninstructed movements. So we started off by looking at how much variance is accounted by the task variables. This quantity here, CVR squared, that's the cross-validated R squared, and that just basically tells you how similar is the model and the data. It's not immediately obvious what to make of these numbers, but of course we can compare them to, for example, instructed movements. And these are the ones that people in the field have been studying for a long time, and that's good because they account for a lot of single trial variance. So well done to the field. But the big surprise here was when we looked at the contribution of uninstructed movements. So these are things that aren't really part of the experiment that we weren't even really thinking about at all. We just kind of included in the model for completeness. There are things like pupil, hind paw, nose, body, whisking, video and video motion energy. And these turned out to account for far more single trial variance than any other variable at all. Certainly much more than the visual stimuli, which was surprising because this is a visual or sometimes auditory task. It was puzzling to us that they accounted for so much more variance compared to the sensory inputs. We were entirely skeptical of this result when we first made this observation, did many controls, the first of which is we took steps to address the fact that a lot of these variables are related to each other. And we did a much more stringent method of assessing the contribution of each one by fitting and refitting the model with and without each variable, so excluding them one at a time. And this tells us the unique contribution of each variable that cannot be explained by any other variable group. And when you look at the data this way, again, the uninstructed movements are the sort of the only ones that reliably survive this test. Even in this very conservative way of assessing their contribution, they are driving activity much more than anything else. Okay, so this paper came out a number of years ago, and some folks who read it came to the conclusion that sort of everything is everywhere. We don't need to even think about cortical areas anymore because just all the signals are widespread. So does this mean everything is everywhere? No, it does not. So here's why. So for example, this shows you the unique contribution, that's the quantity I defined just a moment ago, of the left visual stimulus. And you can see that the pixels that are most critical for explaining that variable are in the contralateral primary visual cortex, exactly where they should be. And similarly, if you look for the variance accounted by nose movements, they're entirely, or not entirely, but largely in the olfactory bulb, just exactly where they should be because they reflect the sensory consequences of the movement. So you might be wondering, well, how can this be? How can it be that movements are accounting for so much variability, but there are still neural areas that respond the way they're supposed to? And I think this movie will make this clear. So in this movie, we fit these separate variable groups, task, instructed and uninstructed, and we have presented them at every moment in time. We fit them separately at each moment in time so you can see how they evolve. This moment here is right after the stimulus comes on. There's a large onset transient in primary visual cortex. And so briefly, the task variable, that's the visual stimulus, accounts for more variance than anything else, certainly more than uninstructed movements. But even a few hundred milliseconds later, the visual stimulus is still on. But even in that moment, the task variable contribution has gone way down and uninstructed movement contribution has gone up and actually accounts for a little bit more variance. So in other words, primary visual cortex, V1, is driven by the visual stimulus, just like we all knew, and also by movement signals. So once we came to this realization, we had really two hypotheses about how vision and movement signals could coexist. And the first hypothesis is called pure selectivity. And that is that if you look at all the neurons within an area, you have some visual neurons and some movement neurons, and those are largely separate populations. The alternative hypothesis is that instead you have a population of neurons with mixed selectivity for both visual signals and movement signals. In other words, you might have some neurons that are purely visual and some that are purely movement, but most of the neurons reflect idiosyncratic combinations of visual and movement signals. So why is this interesting? Certainly, we've known for a long time that neurons might be more modulated by more than one thing. That's not a new observation. But recent work that's been led by Stefano Fusi's lab has pointed out that certain kinds of mixed selectivity are indicative of what he refers to as a high dimensional geometry. That is, when you look at activity in firing rate space, it's high dimensional. And this is important because when you have these high dimensional representations, it gives you a lot of flexibility in how the area might be read out by downstream structures. In other words, you might, each of these points just reflects the joint firing rate activity for a single condition. So you might slice up the space this way if you wanted to decode one parameter. You might slice it up another way if you wanted to decode another parameter, and so on. And this was an idea, before we were thinking about movements and vision, this was an idea that had kind of intrigued my lab for a while. In an older paper, we had previously looked at mixed selectivity in a totally different context. It was the rat task I told you about, where they're making auditory visual decisions in a freely moving context. And we noticed that some neurons occasionally were modulated only by rate. And so it didn't matter whether it was an auditory or a visual rate. They only cared about the rate of the stimulus. We described those as having choice selectivity. Some of them were modulated only by the modality of the stimulus, whether it was auditory or visual. And the vast majority of neurons were modulated by both the animal's choice and the modality of the stimulus. In other words, most of the neurons we were recording had mixed selectivity for modality and choice. So when we looked at all of the activity together here, not in firing rate space, but in kind of condition space, then although we found the occasional pure selectivity neuron that only cared about, this case, the animal's choice, we found some choice neurons that had a little bit of modality selectivity, some that had equal modulation by both. And basically, there's no structure in the population at all. It appears as if these neurons are just idiosyncratically kind of sprinkled with modality selectivity and choice selectivity. It's sort of the opposite hypothesis from specific subpopulations. And so this led us to conclude that neurons in the parietal cortex have mixed selectivity for sensory modality and choice. Old paper from the lab. Now, lest you be worried that this poses a problem for downstream areas that care about the choice or only care about the modality, we found that we were able to take this population of neurons and with a very simple, just weighted sum, generate representations that either were strongly different for choice, or if we took exactly the same neurons and just picked different weights, were highly representative of modality. So in other words, a downstream area, simply by weighting the neural activity in different ways, could decode whatever quantity was needed. That is, if you're a downstream area, you could weight the neurons one way to estimate modality, and then weight the neurons another way to estimate choice. And that's really the key advantage of mixed selectivity. It can feel at first that your neurons are kind of terrifyingly bewildering, right? They mix all these different parameters, but in fact, mixed selectivity allows different quantities to be decoded from a single population. Okay, fast forward to now when we're thinking about these visual and movement signals, we naturally wondered whether, again, mixed selectivity was at play. Because after all, as I just showed you, we can account in primary visual cortex for the single trial variance, both with task variables and with uninstructed movements. But there's a problem. We couldn't tell whether this was an example of mixed selectivity or not, because remember, we measured these neurons with wide field imaging. And in wide field imaging, that reflects the pooled signal of many, many neurons, This is the problem that I alluded to with this technique. And so whether there's mixed selectivity or pure selectivity, the pooled signal will look exactly the same. So we weren't able to distinguish these hypotheses using the wide field imaging approach that had been so useful. So instead, for testing for mixed selectivity for movements and task variables, we needed to go to a single cell metric. So here we took our 2-photon microscope where we can measure single trial activity, and we looked at the activity of 5 different cortical structures: anterolateral motor area, medial motor area, primary somatosensory, retrosplenial, and primary visual cortex. We first verified that these areas had the trial averaged activity that had been reported by other papers. For example, ALM is a motor area. It ramps up before the choice. Retrosplenial and somatosensory responded around the time of the handle grab. And as you know, V1 responded when a visual stimulus was present. So having validated the trial average responses, we once again went to our same old model, right? The one you know from before, the linear encoding model. But this time, rather than predicting the activity of pixels, we tried to predict the activity of single cells on each trial using exactly the same approach. Okay, so how do we get at this question of mixed selectivity using this new data set? So what I'm going to show you here is the cross-validated R-squared. Remember, that's just how good the model is for all of the neurons that we studied. We had a little over 12,000. This gray line tells you how much variance is accounted for by the full model. The reason it has this nice shape is because I've just ordered the neurons in descending order. So the one at the left is fit the best, and the one at the right is fit the worst. Okay, fine. So now I'm going to overlay for each individual neuron the contribution from uninstructed movements, task variables, and instructed movements. One example neuron looks like this. Here's another example neuron. And if we look at all the neurons for the task variables, they look like this. All of the contribution of instructed movements, they look like this. And here's the uninstructed movements. Okay, so there's 2 things to take home from this plot. First one is most important. The black line is almost always above the other 2. And this tells us that in almost every single neuron that we looked at, movements account for the majority of the single trial variance. The other point is the one related to mixed selectivity. You can see that individual neurons are again modulated by idiosyncratic combinations of these different task parameters. So this supports the hypothesis that individual neurons have mixed selectivity for sensory and movement signals. So what would this plot look like if there were no mixed selectivity? Well, just to be super clear, it would look something like this, where you would have separate subpopulations that were modulated by uninstructed movements, task variables, and so on. But that is not what we saw. Instead, the data suggests that these individual neurons have mixed selectivity for sensory and movement signals. And this is a paper that was published in 2019. So having published this paper, we decided to make both the data and the code for the analysis freely available. And this turned out to be a great thing because a lot of other folks were interested in testing out this data, this hypothesis on their data as well. These 2 papers came to exactly the same conclusion. These were both papers focusing on rodent neural activity and found that in the rodents, they replicated our results, mostly in a freely viewing non-task context, but it was still great to see the results reproduced. And then 2 other groups looked at this data from the point of view of non-human primates. So to me, this was really interesting because I think species differences, as I'll tell you more about, can offer a really interesting entry point into understanding mechanisms. So I was really excited that this happened. The first group was a paper co-authored by Tremblay and Testard from the Petrides lab at McGill University in Canada. And they had monkeys doing a visually guided matching task, and they were recording from area 8, which is in the frontal cortex. They used exactly the same approach that we used for their monkey subjects, again, comparing the contribution of these different variable groups. And they found, very similar to us, that uninstructed movements had a big impact on neural activity, even in this cognitive task in a totally different species. This was pretty interesting, but you might be skeptical because, in fact, area 8 is an area that's known to have some movement modulation anyway, so it's perhaps not entirely surprising that they found some modulation, although the similarity to our work, I think, is still striking. Another group, led by Heinrich and Nienborg at the NIH, looked in primate, primary, and secondary visual areas. Just like us, they were set up with a camera that tracked the animal's face and the animal's body, and they did almost the same analysis that we did. Here's their analysis, and I'm just going to walk you through it because they have one plotting convention that's a tiny bit different from ours. So they compared the performance of the full model with task and movements. That's shown in orange. It's a thin orange line. You can barely see it with the task model only. So if movements account for a lot of neural activity, then the green line should be below the orange. But what you're probably seeing there is that you can barely see the orange line at all. And that's because the orange and green are almost entirely overlapping. And that suggests that in this behavior, which is just passive viewing, in these early visual areas in primates, that movements don't really account for much neural single trial variance at all. Now, I should say I'm overstating their results slightly. There are impacts of eye movements that they do see, and there are also some differences between their experiment and ours. Still, it's a clear difference between rodents and primates for sure. So some folks, especially folks in the vision science community, and especially those that work on primates, sort of said, well, monkeys and mice are just different, and I knew it all along. And that, of course, is true. Yes, monkeys and mice are different. That's not really news. But I think to me, this kind of thinking misses an opportunity. And that's because species to species variability can offer us an entry point into understanding mechanism. So for example, when folks were hoping to understand the role of the hippocampus in episodic memory, they turned to birds and they compared the hippocampal volume in food caching birds that need a lot of episodic memory and non-food caching birds that don't. And they found that the hippocampal volume was reliably larger in the food caching birds, pointing to a mechanistic role for the hippocampus in that behavior. There's other examples as well, such as small changes in the opsons that determine whether you're a dichromat, trichromat, or even a quadchromat. And without knowing the species to species differences in color vision, we wouldn't have been able to understand how these tiny molecular changes give rise to precise differences in the kinds of wavelengths that we're able to perceive. So I don't yet know what the mechanistic insight is by observing this difference between rodents and primates, but I think with the right kind of research program, I think we can probably get there. Before I tell you about the sort of approach that's needed, I want to first just address a couple of low-level explanations that could possibly account for the difference between rodents and primates that I've been talking about. Okay, so the first possible explanation is that the specialized nature of primate V1 means that movements could corrupt the readout of visual parameters. Okay, so let's be concrete here. Let's suppose we're talking about area V1, and suppose it's modulated by vision and movements. And you're a downstream area that wants to know, say, the orientation of a grading that's presented to you. Well, you don't necessarily want the movement signals there, right? Because it seems worrisome that they would corrupt the signal, and it might be better off to have pure selectivity for just visual signals. But I think that can't be the right explanation. And the reason is that primary visual cortex in the primate and in any animal already have mixed selectivity. They have mixed selectivity for orientation, spatial frequency, contrast, and a number of other parameters. So if you're a downstream area and you observe a decrement in V1 firing, you don't know whether it came from a change in orientation, spatial frequency, and contrast unless you are already taking advantage of the known mixed selectivity of that area to appropriately weight the inputs and decode from the signal what was the change that actually led to the decrement in firing rate in this example. Now, there's clearly some differences here, right? When we talk about orientation, spatial frequency, they're all parameters that bear on the same dimension, maybe even on the same visual object. And that can feel kind of different from having orientation mixed with, for example, movements. And that's surely a relevant difference. I don't dispute that at all. I'm just saying that if I were a downstream area, I might not care if the mixed selectivity came from orientation and spatial frequency versus orientation and movements, because the problem for me as the downstream area is the same thing. I need to decode what actually happened by skillfully weighting the readout. And that problem doesn't change if you have mixed selectivity for one set of parameters versus another set of parameters. Okay, so I think we can rule out that as a possible explanation. Another one, mixed selectivity for movements in mouse V1 reflects the fact that mice are more olfactory and don't really use vision very much. I hope that I would have convinced you already that this isn't the case because of the nice psychometric functions that mice have, but you might not be convinced because this perhaps just shows that we're able to really train animals to make any kind of sensory motor association, and that in their natural life, they would really prefer not to use vision when they're navigating around in the world. But I'm going to show you a couple of ethological behaviors that I hope will convince you otherwise. This is data that was collected by Jennifer Hoy, who's now at the University of Las Vegas, Nevada. She used to be in Chris Neal's lab at the University of Oregon, and she had mice chasing crickets. She didn't need to train this behavior because it's ethological, and she went to great lengths to demonstrate that this was a visually guided behavior by deafening the animals and showing that performance was unimpaired and so on. Another example is this called the pole descent task. This was developed in Aaron McGee's lab, and he set up kind of an ambiguous visual environment, whereas in which if you use binocular vision, you will go to one platform, a closer one, whereas if you're only using monocular vision, you'll go to a different platform. So these two kind of sensory cues are in conflict, and he found that mice reliably used their binocular vision, so they used their own 3D perception to go to the proper platform, unless of course they had one eye closed, in which case they used the monocular depth cue. So this shows not only do they use vision in an untrained spontaneous task, but they use binocular vision as well. And finally, here's another behavior from Chris Neal's lab. The animal's bobbing its head up and down to get a parallax cue about the size of the gap, how far away the platform is, and then the mouse makes just the right movement to jump across the platform. So I'm hoping that kind of, I think we can rule out this explanation, that I'm hoping as a field, we might kind of let go of this idea that certain critters are dominated by one modality or the other. We all have multiple sensory modalities. We all mostly use them in a combined way when we can, and we all weight those incoming sensory modalities according to their reliability, and we change that weighting in a highly dynamic fashion, even in the time scale of minutes to seconds. So all of us are fundamentally multisensory. This doesn't mean there aren't big differences between primate and rodent visual systems. There are, but if we want to really understand those, I think we have to let go of the idea that some critters are visual and some are not visual. Okay, and finally, movement signals might arise largely from a single cell type. How much time do I have left? I'm going to decide how much detail to go into on this point. 3 minutes or 10 minutes? Okay. I'll say this very quickly then. Maybe one thing we worried about was that these movement signals were largely restricted to a particular cell type, maybe deep layer 5B neurons that project subcortically to the superior colliculus and spinal cord. And indeed, in the work I told you about, we were recording from all excitatory neurons, so we couldn't distinguish neurons with different projection targets. But thanks to a collaboration with my former colleague, Josh Wong, he's not a former colleague. We used to be at the same institution, then we both moved. He helped us to separately measure pyramidal tract neurons and intertelencephalic neurons, two different categories of excitatory neurons. And when we repeated these experiments in these separate cell type specific ways, we were able to see the separate contributions of different types of excitatory neurons. We did not find that these cells are identical. They actually do different things, and that was validated by some cell type specific optogenetics that we did. But in all cell types we looked at, we found that movements had a much higher contribution to single trial variance than any other signals. And this was true in every cell type that we looked at. Future work by Lillian Wilkins in my lab is kind of following up on the sensory properties of these deep layer 5B neurons. So if you're interested in those, you can talk to Lillian. Okay, so I think we've ruled out some of these explanations, and in my final 2 minutes or some number of unspecified minutes, I'm going to argue that a new approach is needed to understand how movements can sculpt neural activity to benefit vision in diverse species. And I think an important feature of most of the work that I've told you about so far is that it's quite distant from an ethological context in a number of ways. And one important way is that animals can't really use their uninstructed movements to alter the visual signal very much. And I think if we want to get to the bottom of this issue, we need to both have new methods for large-scale multi-area recording and also behaviors in which movements can enhance vision. So I'll just mention briefly a new device that was developed by Joao Couto and Max Malin in my lab. This is a device that allows us to implant 6 4-shank neuropixels probes into one hemisphere of a mouse. This allows us to track cortical and subcortical structures and to have large-scale whole brain recordings or multi-area recordings in a way that used to be only possible with wide field imaging, and now we can do with electrophysiology, giving us a lot more temporal precision. The recordings that we make are really large. These implants do not impede the animal's ability to run around its cage, and they're stable for up to a year. So this is the number of simultaneously recorded units for over 360 days. A year is a pretty long time. This is when the trainees went to the Society for Neuroscience meeting. This was Christmas, and the cells kept firing, and they're very stable. Okay. So, and finally, I'll just close on making one last point. I think behaviors in which movements can enhance vision are going to be really interesting ones to study. I gave you one example already, this ethological task, but this one has some limitations, right? These ethological behaviors limit the ability to study threshold level behavior, which is sometimes very important, and also it limits our ability to systematically vary key visual parameters. So we need something that gives us kind of the power of the old psychophysical approach combined with the flexibility to allow the animal's movements to be meaningful. One hint about the sort of behavior that might be appropriate for this is the task from the International Brain Laboratory that I told you about at the very beginning of the task. So remember that on this task, mice use a wheel movement to center a grating to report its spatial position to us as the experimenters. But one thing that I didn't tell you about when I mentioned this task earlier is that if you look at what animals do, the moment that the stimulus comes on, before they make that choice reporting movement, many of the animals wiggle the stimulus around. They move it around. Why is this interesting? Well, if you think about it, those of us in the IBL task, we developed an input for the mouse visual system that from a space time point of view looks something like this. It's just a grating that doesn't change over time. This is a terrible stimulus for the visual system. In fact, without any kind of movements at all, this will quickly become invisible to the animal. So this was sort of an odd choice we made when we did the experimental design. And what did the mice do? They used a combine of wheel position and eye position movements to add spectrotemporal content to the stimulus, making it a better stimulus for their own visual system. Not all mice do this. Some mice make ballistic wheel movements that have no wiggles, but it's pretty easy to find mice that wiggle the wheel quite a lot, hence adding important spectrotemporal content to the stimulus. And some mice, not on high contrast trials, but on low contrast trials, actually have a higher proportion correct as the number of wiggles increases. So this is an example of the sort of behavior that we might be able to use to kind of have it both ways, to have the animals in a setup where we can systematically vary the parameters that we need to, in a way that we need to understand neural circuits, but still giving them an opportunity to discover movements that can benefit their own visual processing. I should add that this particular mouse was trained up by Marcia Taheri in my lab, and Damien Trotter, who's in Alex Huck's lab, has also trained up a marmoset on this task. And I think the cross-species comparison is going to be really important here, and I'm hoping for variability across species, again, because I think that variability can be used as a lever that can be insight into mechanism. Okay, so to wrap up, animals and humans make frequent movements during perceptual and cognitive tasks. In mice, these movements modulate neural activity broadly and in multiple excitatory cell types. And finally, that new frameworks can acknowledge how dynamic interactions with the environment benefit sensory processing and decision-making. Devices that allow large-scale measurements during visually guided behavior will also help. And this shows the Indy and also a mouse and a monkey being friends. So these are the folks in my lab that did the work. We're really grateful to the NIH, the NSF, and also the Simons Foundation for supporting this work. Thanks to you all for coming, and I'd be happy to take any questions. Yeah. Oh, hang on.
So clearly your interest is moving towards more naturalistic behaviors, and that's why you developed the Indy, et cetera. But from a vestibular lab point of view, what do you think you're missing in a head-fixed animal, in a head-fixed preparation?
Yeah, I think missing the vestibular signals is a big problem for sure. And I think the vestibular signals are kind of unique among sensory modalities in that they have been known for a long time to target many, many cortical structures. So I think that would be a particularly interesting sensory signal to study. Also, the very short latency of vestibular signals means that they might interact with other sensory signals in a really cool way, right? Because like the visual latencies are more like 100 milliseconds and the vestibular are, ballpark 20. So yeah, I would love to look at that. And I think we have the right instrumentation to do that because we can include on the Indy a gyroscope. We have those currently on our UCLA mini scopes, but not on the Indy. And yeah, we should add that. It's a great point.
Yeah, good. Thank you.
I should say I would love any trainee questions, by the way. I know we've got some trainees, so if you're a trainee and are holding back, please speak up.
Has any trial been done blocking one eye, vision in one eye, blocking vision in one eye in both monkeys and mice? What are the impacts of blocking vision in one eye, let's say left side and then right side? Would that affect the improve the perception on one side of the brain rather than both?
Yeah, that's a great question. So in the particular experiments from my lab that I talked about, they actually were all designed specifically to have full field visual stimuli so that the overall visual inputs would be changed only minimally by head or eye movements on the part of the animal. So our tasks are mostly designed that closing one eye probably wouldn't have a big impact. But there has been a series of really interesting studies starting quite a long time ago and going all the way up to the pole descent task that I mentioned, where experimenters made use of that technique of closing one eye specifically to prevent binocular vision and to see whether the animal can rely on other kinds of monocular depth cues. And the loss of that binocular vision, even in a mouse where the binocular zone is really small, does have a big effect on their behavior for sure. So I think you're right. Thinking about how the two eyes work together is critical. And I think moving forward, when we're in the right regime, I think we will consider those separate inputs from the two eyes in a meaningful way. Yeah.
Thank you for the fascinating talk, Anne. So I was wondering about the definition of the task relevant features and task irrelevant features, sort of how you sort of set a boundary between the two, because when the animals lick or make a motor decisions, there are also movements of the different muscles that are actually relevant to the task, but they may not be reflected in the low dimensional sort of binary variables that you record or sort of log. So I was wondering sort of how to disentangle those.
Yeah, well, there's I think sort of two key points here. So we worried a lot that maybe the large contribution of uninstructed movements was because they were there to sort of support the instructed ones. Like maybe just to be concrete, maybe when the animal is grabbing the handlebars, they're kind of lunging forward with their body. But we were able to actually rule out that explanation pretty quickly, because when we look at the contributions of each variable group over time, the contribution of the uninstructed movements actually dips around the time of the actual handle grab and the actual lick. So if anything, I think they tend to be suppressed by the instructed movements. Then there's kind of another thornier problem, which your question kind of gets to, which is that these uninstructed movements, they really have two varieties. And one of them are what we call task aligned, in the sense that they are aligned very closely to task events, and others are independent. So an example of that is whisking. So when animals are trained up, this is something that happens with learning. Whisking is not part of the task. We don't train them to whisk, but they have a very stereotyped whisk when the stimulus comes on and another whisk around the time that the reward is delivered. Those are called task aligned movements. And the other kind would be whisks that just occur randomly in time. We do have a way of disentangling those. We basically fit a task only model and then compare the task only model with a model that's the task plus the whisking. And the difference between those two is the task independent movement contribution. Then we fit a single variable model for the whisking, see the total amount of variance that's accounted for, and then subtract off that component to get the task dependent component. So we do have a way of pulling those apart. It's kind of a way that's done through the neural data, which is kind of a funny way of doing it. You can do it through behavioral data as well, but that's maybe another story.
Thank you.
Yeah, absolutely fascinating. I'm out of my depth here, so I'm going to ask a really naive question. It seems that kind of as you move forward more and more, you want to assess more and more brain regions so you get a more complete view. And I'm just wondering if one backs up, that kind of seems to me to be kind of the major issue. How does one know exactly where to record, which regions are actually involved, because everything we know so far, I guess one can do functional MRI in the mice, but then they're not awake behaving and doing these things. But one, so I'm just wondering, and then, especially gets complicated when you start getting into non-human primates and, where there are just so many brain regions and, so much more complex connectivity. I'm wondering, like, do you start with these activation experiments with CFOS and that type of thing to kind of analyze all the regions that are hot. Is the whole brain, lighting up when that happens? I'm just trying to get my head around, obviously I'm aware of decades of cognitive neuroscience trying to localize behaviors to different regions and seeing what things do. But based on the kind of work that you're doing, that would seem woefully inadequate for choosing regions. So I'm just wondering how you're approaching that.
Yeah, I think you're right. And I think that the first thing we learned from our large scale approach from the wide field imaging, which isn't even the whole brain, it's just the cortex. We kind of had two wins. One of them is the one that you said, which is that we could look at lots of areas in the same experiment. And the other one is that we could look at the relationship between areas, like which areas fluctuate together and which fluctuate independently. And both of those are really big wins. I think CFOS is probably not quite the right technique because a lot of these signals are kind of transient and the temporal dynamics are really important. But I think we are benefiting a lot from one of the deliverables from the International Brain Laboratory, which is that we did a whole mouse brain recording with electrophysiology by dividing that task up among many labs. So there were about 8 of us labs where we all committed to doing as many insertions as it took to cover the whole entire mouse brain and get electrophysiological recordings during decision-making of the entire mouse brain. And what you're describing is exactly what happened. Some areas popped up that we knew would be important, like visual cortex and anterolateral motor cortex. And then there were areas that weren't even on our radar, like the midbrain reticular nucleus. Like I hadn't read many papers about that. Turns out to be super important for decision-making. You can decode decision variables and so on. So I think that approach, having that as kind of like a resource now to go back to that brain and say, like, what does this area do during my task is helpful. And also now, I mean, I think in the last 10 years, the magnitude of recordings that we can do in a single animal has just completely transformed. So although we cannot get the whole brain electrophysiologically in an animal, we can get, many thousands of neurons from many areas in a way that was impossible before. So I think we're in a really good position to do these large scale recordings, both to identify new areas and to determine why they're important, and to look at the relationships between the activity in different areas, and finally to see how both of those things change during aging and also in mouse disease models. So I think we're at a really exciting place to be able to characterize the neural activity in healthy animals well, and to also see what the individual differences are across different kinds of critters.
Hi, and thank you for the talk. Sorry if I missed this, but I was wondering about the mixed selectivity and if it's stable across time. So if you replot for the same neuron across days or weeks, is the selectivity similar? Because I was wondering if the downstream brain regions will have to be dynamically reweighting the neuronal contributions to get to the correct information.
Yeah, that's a great question. So as far as we can tell, a lot of these cells do respond fairly similarly day to day. On that original electrophysiology experiment that we did, it was with like in-house tetrodes that can stay at the same place for a long time. And so we did have neurons that we believe to be the same from day to day, and their mixed selectivity didn't really change very much. Still, I think I appreciate that you're taking the point of view of the downstream area, because it does have a big challenge, which is that if it wants to, even if the mixed selectivity is stable, if it wants to decode choice for one particular behavior and decode modality for another particular behavior, then it's left in the difficult position of needing to re-weight the inputs. And that's something that we know that brains must be able to do, because subjects can do the same task, can sort of flexibly use the same stimuli in different ways, I should say. But we don't know how that's done, and I'm not sure what the right experiment is to really get at that. But it's a super important issue, and I'm glad you brought it up. It's one that I hope to understand one day.
Thank you.
Is there one more?
Last question.
Okay.
We used to talk. We used to think about the V1 and the locomotion.
Oh yeah.
As a sort of like gain. That's
Yeah.
what we call it, locomotion gain. So how much information about movement is in the V1 activity? Like, can we tell the right movement versus the left movement, or like it's just we can tell it's moving, like there's a movement?
Yeah, totally great question. And I don't have the answer for that, and I'll tell you why. But I think it's really, really important. So you referred to an earlier literature that was started by Chris Niels and Michael Stryker, where they demonstrated that neurons in V1 fire more when the animal was running. And we really want to know the relationship between those kinds of running movements and the kind of fidgety movements that we see in the lab. And we want to understand also how that is different in mice versus primates, because I think understanding potential differences is really important. That has been a really hard issue to get at. So our first attempt to do this, we did our sort of a task very similar to the ones I talked about, head fixed animal, and we put a treadmill there. We thought, oh, this is going to be easy because when the animal runs, we'll just see if its probability of being correct is higher. It seemed like a very simple way to bring that approach and bring it to like our task context, like to bring the Neil lab to the Churchland lab and blend the two. But what happened is the mice outsmarted us. So here's what they did. On every single trial, when the stimulus came on, they would run. And then when the reward was delivered, they would stop. So we couldn't ever compare trials with and without running because the animals ran on every trial. So we're now starting to come up with different ways to test that. And in fact, Gabriel Rojas Bui in my lab has sort of a workaround that I think is going to start to get to the bottom of that issue. But I think we can now relate the movements themselves, understand how individual movements modulate V1 neurons. The reason we didn't do it yet is because in our original experiments, we just took the raw video and put the video into the model. And this, in a way, was a really smart thing to do because it was kind of a hypothesis-free approach, and it told us something about the brain that we really didn't know about the impact of these movements. The downside, though, is we didn't really know what actually happened, right? Because we just know that the video accounts for a lot of the variance. We can project those pixels back up into the full dimensional space and see where they are in the video. But they're things like, oh, so a bunch of pixels around the jaw and around the eye, and sort of figuring out on a single cell level which movements and which visual stimuli are encoded and how they interact has been harder than we thought. But Lillian Wilkins in my lab is working on that through her imaging, doing large scale imaging in V1. So I hope I'll have an answer. I hope Lillian will have an answer for you soon.
Well, let's thank Anne again for the fascinating talk.
Thanks, everyone.